Hi everyone, welcome to Medical Microbiology. So today we will continue chapter 12. This is going to be the last lecture, which is lecture number 5. We will talk about the drug resistance bacteria. We're going to talk about the tests for effectiveness of the drugs, and also we will discuss a little bit about MIC, minimum inhibition concentrations, and TI, therapeutic index of drugs. So we start with the drugs resistance. Okay, so remember the ideal drugs should be the drugs that will not will not cause the bacteria to be resistant to the drugs. Okay, so the bacteria that resistant to drug is mean that the bacteria is actually stronger, and then in order to kill them, then we have to increase the dose of the drugs. Okay, so now the question is how these bacteria are able to be resistant. We're gonna see a little bit about how they actually have the resistant gene. Again, remember when we talk about the structure of bacteria, okay, in chapter 4 and 5, okay, when we talk about the prokaryotic cell, remember there are some bacteria that actually have the pilus, or pili, which is the channel that can be used to transfer gene to other bacteria. Okay, so this is the what we call the pilus or pili, which is the channel used to transfer something from one bacteria to another bacteria. So many gram-negative bacteria like E. coli, Pseudomonas, for example, they have the ability to transfer, let's say this is a gene, special gene is called the resistant gene. Okay, this resistant gene actually can be transferred into another bacteria over here. And what happened, this bacteria then is become resistant. Okay, so this is a natural ability of some bacteria that are able to transfer resistant genes. So E. coli, for example, they're able to transfer the resistant gene to another gram-negative bacteria like Bacteroides, for example, Neisseria, Shigella, Klebsiella, so Serratia, for example, are able to transfer the gene to E. coli. E. coli are able to transfer the gene to the Pseudomonas aeruginosa, or Pseudomonas able to transfer the gene to E. coli. So you see this one? Okay, so this is how they able to transfer the resistant gene to many bacteria. They are also able to transfer cross groups. So this group over here is actually gram positive. Okay, so this group over here is gram negative bacteria. So mostly gram negative bacteria are able to transfer the gene between them. And they're also able to transfer the gene to the gram positive bacteria, like gram positive bacteria. For example, E. coli, they're able to transfer to Bacillus, or Staphylococcus aureus. This is gram positive to another gram positive bacteria. So this is a natural kind of process that some bacteria have the ability to transfer their resistant gene, sharing their resistant gene to another bacteria. Now, the mechanism that they perform. So if that bacteria, for example, have the resistant genes, they usually have the ability to perform this process. Okay, for example, Staphylococcus aureus, yeah, they have the ability to inactivate the drugs, penicillin. So inactivate the drug. So they have or they produce an enzyme is called penicillinase. So this resistant gene produce the penicillinase, and this penicillinase is actually will destroy or block the penicillin. So when we use penicillin to this bacteria, then it will not be effective because it will be broken down by the penicillinase. So example of bacteria that produce penicillinase is Staphylococcus aureus. So Staph aureus, okay, this bacteria actually have the resistances against penicillin. So it's mostly penicillin will be not effective against Staphylococcus aureus. So this is the first process that may be happening in bacteria that have resistance against drugs. Another process may be by decreasing the permeability of the drug. It means that some bacteria that have resistant gene, they're able to produce proteins that will block the entrance of the drugs. So, for example, if we take the drugs, then this drug will not enter into the bacterial cell because of this action. Okay? The next action will be increase the eliminations of drugs from the cell. Okay? So again, some bacteria that have resistant gene, the drugs will be enter into their cell. Okay, so let's say this is a drug enter into the cell, but this bacteria is actually have the ability to pump this drug out from the cell. Right? So increase the elimination of this drug from the cell. So therefore, these drugs will not be effective against this type of bacteria that have the ability to remove the drug from the cell. Or there are some bacteria that have the ability to produce receptors that will not recognize the drugs. So for example, let's say this is a bacterial cell. Usually there is a receptor that will receive the drugs. So if this bacteria have this receptor, then this bacteria will be mostly killed by the drugs. Okay, but there are some bacteria that produce another type of receptor that will not bind the drugs because different shape, for example, like this. This one is curved. It will be. So when this bacteria have the ability to produce this receptor type, then the drugs will not be able to bind to the receptor, and this bacteria will be able to protect themselves against the drugs. Okay, so they change the receptor. So if they use to have this kind of receptor, then they change it. They make another type of receptor that is safe from binding the drugs. And the last action will be there are some bacteria, if they do have the resistant gene, they're able to change their metabolic patterns. Remember, sulfa drugs, they're able to compete with the substrate in order to block the metabolic pathway. So if the bacteria has the resistant gene, they able to change that pathway. Therefore, sulfa drug, for example, will not be effective against this type of bacteria that able to change their metabolic pathway. Okay, we're gonna see the picture of that activity. Okay? Again, the first action may be drugs inactivation. Okay? So this is penicillin, like this. So if bacteria are able to produce penicillinase, this penicillinase actually will break this penicillin into a type that is inactive against the bacteria. So bacteria will be resistant against penicillin because it produces penicillinase that break the penicillin. The second action again is to decrease the permeability. It means that the drugs will not be able to enter into the cell. Right? So this one is the drugs able enter into the cell, but because it's this bacteria able to decrease the permeability of the drugs, that most drug will be outside the cell and this bacteria will be safe. And the next one will be the again the activations of pumping the drug out, okay? Removal of drug from the cell. So let's say the drug is actually enter into the cell. However, this cell, the one that is resistant, has the special pump over here that will pump the drug out. So finally, the drug actually will not stay in the bacterial cell. So this bacteria become resistant against this drug. The next action is by changing the binding site. So just remember, for example, there are some drugs that bind into the smaller subunit or bigger subunit over here. So if the bacteria that have resistant gene, they're able to change this receptor. So these drugs over here is actually will not be able to bind to the bacteria that able to change the receptor on the ribosome. So this drug actually will not bind to the ribosome. So the bacteria will be protected. And the last one is the bacteria that have the ability to change their metabolic pathway. For example, this is the normal metabolic pathway, and then if you put the sulfate drugs over here, then there will be no product. Okay. Now, this bacteria has the ability to change this pathway. Instead of from B into C, they change it into B into C1. And when they change the metabolic pathway from B to C1, these drugs will not be effective against this new metabolic pathway. So basically, then the bacteria will be safe. They will be able to produce their main products. So these are the mechanism of resistance, okay, bacterial resistance against drugs. There is another resistance with the process what we call natural selection. Okay, so just remember, in one populations, let's say human populations, we have mostly a regular type of people, right? But there are also some people that is actually not a very they have special condition. Maybe they are stronger compared to just regular people. So the same thing with this bacteria in population, mostly will be just regular type of bacteria, but there are some of bacteria that's actually really strong. Okay, so this bacteria is actually resistant against drugs. Okay, let's say we put drugs into this population, boom, and then what happened? Mostly the regular bacteria will be killed, and then only the strongest bacteria stay. And then after several weeks or months, for example, the one that develop inside the cell will be mostly the resistant bacteria. Okay, and then this will be growing as a new population over here. And these resistant bacteria, actually, there are some of them will be also even stronger. Okay, so this one is become regular now. So in order to kill this stronger bacteria over here, resistant bacteria, then we need to increase the dose, so higher dose of drugs. And this higher dose of drugs will mostly kill the resistant bacteria over here, but not killing the very, very resistant bacteria. And what happened then, this bacteria is the one that will become the new population. And in order to kill this new population, we need the even higher dose of infection. So this is what we call the natural selection of drug resistance in bacterial population. Now, this is a very important for us and also for physicians and government. So when we use antibiotics or drugs, so physician has to be sure that they're giving the right prescription, the right drug, the correct drug that will kill or cure the infection. So the physician will not give the drugs, kind of trials drugs to the patient. Otherwise, it will cause the infection become stronger, or the bacteria become resistant. And for us as a patient, this is really important that we have to comply with the rule. So like, for example, if a doctor telling us to finish the drug within 10 days, then finish it in 10 days. So do not stop taking that drug. For example, maybe in the 5th day, we already feel better. We think that we are better. It is actually not, because not all bacteria actually kill in 5 days. There are still some strong bacteria still alive. So if we stop taking the drugs in the 5th day, then what happens? The strong population will be growing inside our cell. And if we need to kill them later, then we need higher dose. So if the doctor say finish the drug within 10 days, then finish it, all the drugs that, doctor give to us. Otherwise, if we just use half of them, then there will be a resistance. The strong bacteria will still be reproduce or replicate inside our cell. So administrations drugs, is also to be careful. So there are some drugs is broad spectrum or narrow spectrum. If the doctor or the pharmacy already know that the infection is really special or specific, is only one bacteria that causes infection, it is better to just give the narrow spectrum of drug that only kill that bacteria. Because if we use the broad spectrum, it might kill other bacteria that may be efficient for us, benefit to us. So it's really important, again, the drug administration to give the correct drug. The best drug will be the narrow spectrum if the infection is only one type of infection. If it is mixed infection, then it can be given the broad spectrum of drug. So for the pharmaceutical companies, so drug research, for example, they need to focus with the drug that have a short-term effect. It means that it's not causing the resistance to the drugs, because it's long-term use. It might increase the capability of the bacteria to be resistant against that long-term type of antibiotics or drugs. And then long-term strategy, this is mostly for government. For example, there should be rule of using the drugs, the antibiotics. Sometimes farmers, for example, they also use the antibiotics to their cattle. So if they do not use correctly, so the bacteria inside the cattle is actually become stronger and stronger, and it can jump to give the infection to human, for example. and this, the one that give infection to human, will be stronger type of bacteria because they already resistant against the drugs that is given to the cattle, for example. Okay. There are some people that actually have a side effects against the drugs, so but 5% mostly drugs can cause side effects, especially to some people that have kind of lower immunity or they have some problem with their body. They might develop the side effects. For example, maybe the side effect could be the direct damage to the tissue due to the toxicity of the drugs. So like for example, HIV drug usually causing this problem to the user. Okay. Allergic reaction. Some people have allergic reaction against penicillin, for example, or maybe disruption of balance of the normal flora. And when it is happen, that it can cause what we call the super infection. So what is the example of this super infection? Let's say we have the infection in our throat. Okay, so we give the drug. Let's say we give vancomycin. Okay, vancomycin usually used for throat, like strep throat infection. So this vancomycin actually will kill this bacteria over here, but it also going down to the GI system. And finally here in the intestine, these drugs able to kill the natural flora. So indigenous flora or natural flora that is important for us. This is what we call the probiotics. Okay, so this is the one that important for us. This natural flora actually compete against the pathogenic flora over here. Okay? This pathogenic flora will not cause infection to us because they compete with this natural flora. So this natural flora actually keep this pathogenic flora in balance. So when this drug enter into this system, what happened? It kill the natural flora because maybe natural flora is more sensitive against these antibiotics. And what happened? This one is growing. And finally, the one that's growing in the intestine is pathogenic flora, and then it causes the disease. So this is what we call the super infection, which is the infection in the site due to the use of antibiotics on another site. So the infection that happening in this intestine is called the super infection. This is the idea why people, for example, suggest to take the probiotic in order to grow more natural flora. You can read this in your textbooks. This is some major side effects on human body. Okay, like for example, penicillin might have a side effect on the skin, like rash, itchy. Tetracycline, if I hear, for example, it might have effect on discolorations of teeth. So the teeth become yellow for longer use of tetracycline, for example. So this is again some list of side effects on our body caused by the antibiotics. Okay, this is consideration of selecting the antimicrobial drugs. So usually there is a test to know whether the drug is effective or not, and what is the best concentrations of the drugs. Okay, what is the best dose for the drugs in order to cure the infection. There are several types, okay, of identification. Okay, like for example, to test whether the drug is effective or not, usually using what we call the Kirby-Bauer diffusion test. Okay, so this is usually for effectiveness. Effectiveness of drugs. MIC, for example, over here, this is usually to know the dose of drugs. Okay, so what is the best dose used for making the drugs that will kill or destroy the infection? Okay, this is the Kirby-Bauer test, which is the diffusion test in order to test the susceptibility or sensitivity of drugs against bacteria. So this different color over here is actually, this is bacteria growth. Okay, this is bacteria growth. Okay, so when we put the drugs or antibiotics on this bacteria population, like say for example here, okay, chloramphenicals, this is ampicillin, okay, gentamicin, for example, enrofloxacin, oxytetracycline, cefotaxime. Okay, so 1, 2, 3, 4, 5, 6. So 6 different drugs is actually used for this test. Okay, let's see what type of bacteria they use. It doesn't say, but let's say this is used on Staph aureus, Staphylococcus aureus. Okay, so this is actually a lot of Staphylococcus aureus growing in this petri dish, right? So when we put the drugs in it, and what happened, there is a zone of inhibition over here. Okay, so this is a clear zone, okay, or zone of inhibition. Okay. What does this mean? In this clear zone over here, okay, there are no bacterial growth. Because of what? Because this drug actually kill bacteria in this area. So you see this one, this area is clear, this area is clear, clear area, right? This clear area is mean that there is no bacterial growth. So we can measure this clear area, you know, by using ruler, for example. So the wider the area is mean that the more effective the drug's gonna be. Like for example, this enrofloxacin actually the most effective drugs against this. Let's say this is Staphylococcus aureus because it has wider zone of inhibition around 10, 20, 25, 26, 27, okay, 27 millimeter wide. Is really effective compared to this one, for example. Okay? So oxytetracycline is only maybe what, maybe 10 millimeter. So this is the least effective one. This one may, you know, the next one that is effective because it is also wide. This one may be the third effective. This is maybe the fourth effective. It's almost similar. Okay? This is the fifth effective one, and this is the least effective type of drugs. So this is what we call the Kirby-Bauer diffusion test to test the effectiveness of antibiotics against certain type of bacteria. So we can change this bacteria. Maybe we use E. coli, for example, or we can use another type of bacteria to check, to know which antibiotic is actually more effective against this type of bacteria. So based on this, then, you know, when the doctor give the prescription, then the doctor will give this one over here because this is the most effective one. The doctor will not give this one, because this is very, very less effective. Okay. This is the test for identifying the dose that is given to the drug. It's called the MIC. Okay, MIC, which is minimum concentrations, minimum inhibitions concentrations. Okay, there's the lowest concentration that inhibits bacterial growth. So minimum inhibition concentration. Now, how the test done? This is by, so this tube is actually have bacteria on it. So before this tube given the drugs, okay, so the drugs will be put into this tube. So this tube actually has bacterial growth, like the control over here. So all of them start with this, this color, all of them. Now, the control, we will not put any drugs in the control, so zero concentration. On the first tube, okay, after the control, then we put, let's say, 0.2 dose, milligram per liter, for example. And then on the next tube, we double the concentration, 0.4. We put it over here. And then we double 0.8, double 1.6, double 3.2, double concentration 6.4, double the concentration 12.8. If we have another tube over here, then it's going to be around 26 25.6 dose. Okay. So we put that drugs with this concentration. What happened after several days, maybe 1 day or 2 days, we check what happened. So basically in the control, because there is no drugs, the bacteria will grow. So you will see this bacteria over here, okay, so cloudy. And 0.2, maybe still bacterial growing, almost similar with the control. The next one still growing, but a little bit less, and then this one a little bit less, a little bit less, this is a little bit less, and finally over here, no bacterial growth. It means that bacteria will be killed by this 6.4 dose. And then of course, the 12.8 will be no bacterial growth because this is higher dose. So based on this information, then we know this is actually the minimum concentration that will kill the bacteria. So the MIC from this test will be 0.4 milligram per liter. So 0.4 dose will be the one that will be used, for example, for development of drugs with this dose. Okay, so this is how to test for the MIC. Okay, so remember MIC, this is done outside human body. It's called the in vitro test. Okay, so what happens if it is in vivo? It's maybe a little bit different. Therefore, we have to know whether that drugs, if it is given to human body, will be safe or not by calculating the what we call the therapeutic index. So we basically have to also perform the test inside the cell, inside the human body. Let's say, after the test, we perform the MIC test. Let's say we have 3 different drugs, maybe A, B, C. Okay, so this drugs is used for this, the same type of infection. Let's say when we test the MIC, okay, for drug A, let's say we have 1 milligram per liter. Okay, the MIC is 1 milligram per liter. And when we test B, it's actually also 1 milligram per liter. And C also 1 milligram per liter. Okay? So this is the MIC. And then we also perform toxicity tests to human body. So toxicity tests. Okay. Let's say drugs A actually causing toxicity when it is reaching 10 milligram per liter. Okay, drug B, it causing toxicity when it is reaching 1000 milligram per liter. Okay, it means that it is safer because it's given toxicity to human body at higher concentration. Let's say C is actually giving the toxicity to human cell at 100 milligram per liter. Okay, so based on this test, the MIC test and toxicity test, we actually able to calculate the TI, okay, the therapeutic index. Okay, so the TI for A will be, so we actually divide the toxicity against the MIC. So A will be 10 divided by 1. It's going to be 10. So the index will be 10. B will be 1000 divided by 1, by the MIC. And then the result will be 1000. And C is going to be 100 divided by the MIC, which is 1. Then the result will be 100 Ti. Now, from this information, remember the high index is desirable because high index means it's actually safer. So based on this information, then the best drug to be used will be the B antibiotics, okay, the B drugs, because what? The TI is 1000. So the higher the index, the better the drug's gonna be, more efficient and also safer to human body. I think that's all for chapter 12. We finished chapter 12. We will see you again on the next chapter, which is chapter 13. Goodbye now.